Dopamine is making a decision inside your brain right now. Not pleasure, not happiness, a decision, and you have no idea it is happening. Dopamine decided what you ate for breakfast, whether you hit snooze or got up, whether you kept scrolling last night or finally put your phone down. Approximately 400,000 neurons power 4 pathways, one molecule controlling motivation, movement, and every choice you make. You scroll for 3 hours, you do not enjoy a single minute, you just cannot stop. That pull you feel is not weakness, that is architecture. Every scroll triggers a tiny dopamine pulse in your nucleus accumbens, not a flood, a whisper, just enough to keep your thumb moving to the next one. Systematically, over 28 minutes, you will see exactly what this molecule does in your brain, every pathway, every receptor, every trap. In 1993, neuroscientist Wolfram Schultz placed a tiny electrode near a single dopamine neuron in a monkey's midbrain. Then he gave the monkey a squirt of juice. Unexpected juice, the neuron erupted in a firework of dopamine. Schultz recorded 20 to 30 pulses per second, the highest burst he had ever measured. Repeatedly, Schultz ran the experiment day after day. Within weeks, something remarkable happened. The neurons stopped firing at the juice. The neuron moved. It stopped caring about the juice and started firing at the bell, the signal predicting the juice. The brain learned to anticipate. That discovery shattered the pleasure theory. Dopamine encodes the gap between what you expected and what you got. Neuroscientists call this reward prediction error. Prediction error changed neuroscience forever. Understanding your dopamine system starts with what happens at baseline before the first sip of coffee. Before your alarm rings, before your first thought forms, your ventral tegmental area is already humming, 3 to 5 electrical pulses per second, a quiet, steady rhythm. Underlying this calm rhythm is tonic firing the baseline. Think of it as an engine idling, the neurochemical hum that gets you out of bed. Dopamine releases in tiny doses and gets recycled within 200 milliseconds, a steady signal that says, you are alive, you can choose. Dopamine starts as tyrosine, a common amino acid. Tyrosine hydroxylase converts it to L-DOPA, the bottleneck of your entire motivation system. The AADC enzyme finishes the job in milliseconds. Fresh dopamine gets packaged into vesicles and shipped along the axon to wherever it is needed. Remarkably, 4 pathways carry dopamine to radically different destinations: reward, thinking, movement, hormones, same molecule, 4 separate jobs. 6:45. Your alarm fires. The nigrostriatal pathway sends dopamine to your striatum to move your arm. That takes 80% of your brain's dopamine. Olfactory signals reach your VTA, and the mesolimbic pathway brightens. Not because coffee is pleasurable yet, because your brain predicts it will be. Essentially, baseline dopamine is your engine idle. It keeps the system running. Something is about to shift that hum into a roar. A Stanford psychiatrist called your phone a modern-day hypodermic needle for digital dopamine. The baseline system you just learned your phone is constantly tapping it. Everything so far has been tonic firing the steady baseline. When something unexpected arrives, dopamine shifts to phasic firing, a completely different signal. Contrasting tonic hum with phasic burst reveals one of the most important discoveries in neuroscience. It starts with a monkey and some juice. Phase 1. Unexpected reward. Juice arrives with no warning. The dopamine neuron explodes 20 to 30 pulses per second, a positive prediction error. Prediction emerges over repeated trials. A bell rings before the juice. Within weeks, the dopamine burst migrates from reward to bell, firing at prediction. Finally, the bell rings, but no juice comes. The neuron drops below baseline, a negative prediction error, the brain's signal for disappointment. First bite of chocolate. Your nucleus accumbens blazes with a massive dopamine burst, a positive prediction error. Your brain tags this experience as worth repeating. Identical chocolate, identical taste buds, but your dopamine neurons go quiet by bite 10. The reward was predicted. Prediction error reaches 0. 0 means no signal. Dopamine does not reward you for getting what you want. It fires when reality beats expectations. Your entire reward system runs on surprise. Neuroscientist Kent Berridge removed all dopamine from a rat's brain. The rat could see its food 3 inches away. It did not walk over. Researchers placed food directly in its mouth, and the rat still enjoyed eating. Pleasure was intact. Desire was gone. Liking without wanting, 2 separate systems. Dopamine drives wanting. Opioids drive liking. Your brain can make you chase something you no longer enjoy. Remember that. Reward prediction error is not just biology. Schultz's discovery matches temporal difference, learning the same algorithm at the core of artificial intelligence. Simultaneously, thousands of neurons across your VTA run these predictions in parallel. Every experience is compared against what your brain expected. Prediction errors feed directly into your prefrontal cortex, the region that weighs options and makes every decision you face. Prediction feeds into decision. Decisions happen here, the basal ganglia. 2 pathways diverge at a neural fork. One pushes action, the other holds you back. Dopamine arrives and tips the fork. D1 receptors on the direct pathway light up green go, D2 receptors on the indirect pathway dim red stop. Dopamine tips the signal inside your skull green for go, red for stop. Level 3 begins how dopamine architects every decision. D1 receptors activate the direct pathway, green light, saying go, pursue, engage. D2 receptors activate the indirect pathway, red light, saying wait, suppress, conserve. Directly from striatum to thalamus to motor cortex, a green cascade promoting action. Indirectly through a longer red circuit that blocks the competing choice. Gymnasium or couch the fork computes, dopamine biases the D1 pathway just enough. Green wins. A decision made at the molecular level. A 2020 study in Science gave participants methylphenidate, a dopamine reuptake inhibitor. It did not make tasks easier. It changed how their brains perceived effort. Dopamine does not increase your ability, it increases willingness. Additional dopamine in the striatum makes effort feel less costly and reward feel more likely. Perfectly calibrated, refined by millions of years of evolution. 3 pathways explored, but nobody tells you what happens when something overwhelms it. Your prefrontal cortex holds options in working memory comparing, weighing. Dopamine keeps those circuits active. Without it, options flicker and fade before you decide. When dopamine supply drops to this region, options dissolve before you can compare them. Decisions feel overwhelming the circuit cannot complete. Elegantly balanced when calibrated, refined by millions of years of evolution. Something floods it with 10 times the normal signal. Everything breaks. Food, 150% above baseline. Sex, 200. Cocaine, 350. Methamphetamine, 1000. The scale is overwhelming. Concentrations this extreme drown the synapse entirely. The DAT transporters that normally recycle dopamine in milliseconds are overwhelmed, blocked, or reversed. Everything you spend 3 levels understanding is about to be hijacked. Addiction is not a moral failure, it is an engineering problem. Under normal conditions, the dopamine transporter recycles dopamine into the neuron within milliseconds. Cocaine enters the synapse and heads straight for this transporter. Cocaine wedges into the transporter like a key jammed in a lock. The door shuts. Dopamine accumulates 350% above baseline. Amphetamines reverse the transporter entirely. Instead of recycling dopamine inward, the DAT now pumps it outward, a molecular fire hose. Flooding the synapse repeatedly triggers a counterattack. D2 receptors retract into the membrane, pulled under like fishing poles, 25% fewer within weeks. Ordinary dopamine signals barely register against the depleted membrane. Food that once tasted incredible now feels bland. Music that moved you sounds flat. Tolerance and sensitization create a paradox. Wanting grows louder, while liking fades to a whisper. Chasing harder. Feeling less addiction's neural signature. Every spike of pleasure is counterbalanced by an equal pull toward pain. Small spike, small rebound, the brain resets quickly. Massive spike, massive crash. Repeating the cycle warps the seesaw permanently, diminishing pleasure each time, deepening pain between hits. Eventually, the substance is needed to feel normal. Addiction is a medical condition, not a character flaw. If someone is struggling, seek help. The mechanism is not the person. The same dopamine circuits that respond to drugs also respond to ordinary triggers. Your phone delivers microdoses of anticipation hundreds of times every day. Pharmaceuticals produce a tidal wave. Phones produce a drip. But a drip that never stops can erode the same receptors over months that drugs erode in weeks. Perpetual microstimulation replaces the tidal wave. Level 5, how your phone, your feed, and your food exploit the same ancient circuitry. Slot machines work because you cannot predict the next win. Your social media feed uses the same principal variable ratio reinforcement. Every scroll is a pull. Dopamine fires not when you find something interesting, but the moment scrolling begins. Possible reward activates the prediction signal, weaponized by design. Collectively, the prefrontal cortex, your brake pedal, is outcompeted. Architecture tilts the impulse to scroll faster than the impulse to stop. Ultra-processed food is engineered to hit what scientists call the bliss point, the precise ratio of sugar, fat, and salt that maximizes your dopamine response. Natural food produces a gentle dopamine curve, slow rise, sustained plateau, soft landing. Processed food produces a spike, a crash, and a craving for another round. Adolescent brains are especially vulnerable. The reward circuit is fully online, but the prefrontal brake system remains under construction until the mid-20s. 14,000 upvotes for someone writing day zero of changing their life. The instinct behind a dopamine detox is real, but the name is scientifically wrong. Reducing overstimulation allows D2 receptors to regrow over weeks. You cannot detox from a molecule your brain produces. Recalibration is the correct word. Recalibration works for the overstimulated, but for some people, the dopamine system breaks from disease, and that is a very different story. The substantia nigra dopamine neurons controlling movement. In Parkinson's disease, these neurons begin dying, not suddenly, one by one, like streetlights going dark. Surviving neurons compensate firing faster, producing more dopamine to fill the gap. It works for years. When 60 to 80% are gone, the system collapses. Observable symptoms appear only after the majority of neurons are gone. The brain masked the damage for years. This is level 6. Schizophrenia floods the mesolimbic pathway too much dopamine, producing hallucinations and delusions. The mesocortical pathway is also starved, blunting thought and emotion. Excessive mesolimbic dopamine assigns meaning to noise. Random neural activity gets flagged as significant and real voices, patterns the brain trusts completely. Antipsychotic drugs block D2 receptors to calm the mesolimbic flood, but they also reduce the already low mesocortical signal, worsening the other problem. In ADHD, the prefrontal cortex receives dopamine in inconsistent bursts rather than a steady supply. Working memory slots flicker on and off, not laziness, but circuitry. Medication partially blocks DAT transporters, not flooding the brain, but restoring the minimum dopamine the prefrontal cortex needs. Like glasses they never knew they needed. Parkinson's, schizophrenia, ADHD, 3 different failures in the same dopamine system. Circuit failures, not character flaws. Science is racing to fix them. Researchers just transplanted lab-grown dopamine neurons into Parkinson's patients. The cells survived, connected, and began producing dopamine. Phase 3 trials are running. Progressing from baseline to prediction to decision to hijack to trap to disease and now restoration. Level 7. The Reset Protocol. Returning to the VTA. 400,000 neurons pulsing at baseline. Understanding what that rhythm encodes, working with it, not against it. 14-degree water. Schrammeck and colleagues measured the dopamine response, up to 250% above baseline. Not a spike, a sustained rise. Unlike a drug spike that crashes in minutes, cold exposure produced a sustained elevation lasting 2 to 3 hours, gradual rise, plateau, and slow return. That was a single study with a small sample. Individual responses vary. Cold exposure cannot replace medication, but the profile warrants attention. Regular aerobic exercise adds D2 receptors in the striatum over 8 to 12 weeks. New receptors growing on the membrane measurable on brain imaging. Overnight, in 7 to 9 hours of sleep, the brain performs receptor maintenance, restoring sensitivity on the membrane. Sleep deprivation defers it entirely. 3 evidence-based interventions. Cold exposure for sustained release. Exercise for receptor regrowth. Adequate sleep for nightly restoration. You cannot detox from a molecule your neurons need to function, but reducing chronic overstimulation for extended periods allows receptor density to recover. Receptor resensitization takes weeks, not a weekend off screens. Measurable improvement appears at 2 to 4 weeks of consistent recalibration. Recalibration does not return you to factory settings, but a brain with restored receptor sensitivity responds to natural rewards again. Prediction error is not just a vulnerability, it is a tool. Your dopamine system rewards novelty, routine dulls the signal, variability sustains it. Building surprise into your routine keeps the prediction machine engaged. A different route, a new challenge variability that prevents the signal from flattening. Optimal dopamine is not constant elevation. It is the oscillation between rest and engagement, between prediction and surprise. A rhythm, not a state. Bemdane ProCell stem cell-derived dopamine neurons transplanted directly into the putamen. In the Phase 1 trial, the cells survived at 18 months. Gradually, over months, transplanted neurons extended axons and formed new connections. Phase 3 clinical trials for Parkinson's patients are now actively recruiting. A 2025 discovery revealed that dopamine operates with surgical precision, delivering targeted messages to specific nerve branches at exact moments. One molecule, 7 levels. From the quiet hum of baseline firing to prediction errors, decisions, hijack, modern traps, disease, and restoration. Dopamine is not your enemy, it is your operating system. Your brain is not broken, it is running software designed for a different world. Remember Schultz's monkey from the beginning? The neuron that stopped firing at the juice and started firing at the bell? Understanding changes everything. Tomorrow morning, alarm fires, nigrostriatal pathway moves your hand, coffee smell, mesolimbic prediction fires. Every moment now has a neural explanation. Morning cold exposure sustains dopamine for hours. Midday exercise with variability engages prediction. Evening wind down supports receptor maintenance during sleep. Understanding the system is the first step toward maintaining its rhythm. You cannot control the molecule, but you can respect the machine. 400,000 neurons, the same ones from 25 minutes ago, still firing at 3 to 5 pulses per second, still predicting, still choosing. Anhedonia, compulsive behavior, attention struggles, movement changes. If anything here described your experience, please speak with a healthcare provider. Dopamine, not the pleasure molecule, not the enemy. The prediction engine, the decision architect, the ancient signal asking, what comes next? Stem cell-derived dopamine neurons are not theoretical. Phase 3 trials for Parkinson's are actively enrolling. The nigrostriatal pathway may one day regrow. Precision medicine targeting specific dopamine pathways, restoring movement without triggering compulsion, stabilizing attention without blunting personality. Every morning, the system resets. Every prediction error teaches, every decision passes through the fork. Your understanding of the molecule just changed. 28 minutes ago, this molecule was a stranger. You knew the name, maybe the myth, the pleasure chemical. Now you know the architecture. Every brain watching this runs the same architecture. Different baselines, different vulnerabilities, but the same 4 pathways and the same prediction engine. Understanding is the intervention. The molecule is not the enemy. The system is ancient, precise, and astonishingly well designed. Respect it. Dopamine was one chapter. The next video in this series covers a molecule that controls something you probably never think about consciously. Consider subscribing to Healthicore if this changed how you understand your brain. The next episode explores a system most people never realize they are running. Everything you learned, prediction errors, receptor dynamics, the decision fork started with one molecule and one monkey. Remember that. Dopamine, synthesized from tyrosine, 4 pathways, 5 receptor types, 400,000 neurons, one molecule beneath every decision you will ever make. References for everything in this video are linked in the description and pinned comment. 40 peer-reviewed sources. Understanding your brain deserves that rigor. Healthicore, understanding the systems that run you, from the molecule up. Thank you for 28 minutes. Your dopamine system noticed.